Hello, my name is Charmaine Espinosa, and I'll be presenting the results of the experiment we have conducted at the University of Illinois, which is the effect of a novel consensus bacterial 6 phytase variant on mineral digestibility and bone ash in young growing pigs. A traditional U.S. swine diet mainly consists of corn and soybean meal, and these feed ingredients contain phytate, which consists of 6 phosphate molecules bound to a myo-inositol ring. The problem with that is pigs do not synthesize adequate amounts of endogenous phytase to liberate the phosphorus that is bound to phytate. Thus, majority of phosphorus in plant sources is not available for absorption and is the reason for the low digestibility of calcium and phosphorus by pigs. Therefore, microbial phytase is usually included in diets for pigs to increase phosphorus absorption and utilization by hydrolyzing phytic acid within the gastrointestinal tract of pigs. However, pig diets may include feed ingredients such as canola meal and rice bran due to the availability of these feed ingredients in some areas. Corn and soybean meal contain 0.5 to 1.7% phytate, and these concentrations are less compared with canola meal and rice bran that typically contain 3 to 8% phytic acid. Therefore, if we include a significant portion of these feed ingredients in the diet, it is expected that the concentration of phytate will increase in the diets. With that in mind, we wanted to determine if supplementation of high doses of phytase is needed to reduce the negative impact of high concentration of phytate in the diets. High dose of phytase is hypothesized to increase degradation of phytate in the intestinal tract of pigs and therefore increases the release of nutrients other than calcium and phosphorus. Phytic acid is unstable in the free acid form and occurs mainly as a complex with metal cations. Thereby, the formation of these complexes reduces the digestibility of minerals in the gastrointestinal tract of pigs. Therefore, the use of high dose of phytase in diets may exert positive effects on nutrient digestibility due to increased reduction of the anti-nutritional effects of phytate. However, the effect of phytase in high phytate-containing diets are limited. A next-generation biosynthetic bacterial 6 phytase has been developed, and it is believed to increase phosphorus availability in diets for pigs. In an in vitro study, it was demonstrated that this novel consensus phytase has high thermostability and also has high relative activity over a wide pH range. However, there are limited data to demonstrate the efficacy of this phytase. Therefore, an experiment was conducted to test the hypothesis that the negative impact of phytate is reduced at higher phytase doses. It was also the objective of this research to test the hypothesis that inclusion of the novel phytase to diet increases bone ash and apparent whole tract digestibility of minerals in diets containing varying phytate concentrations. And lastly, to test the hypothesis that the novel phytase is as effective as the Butyroacella phytase in exerting positive effects on nutrient digestibility and bone ash in young growing pigs. For this experiment, a total of 18 diets were prepared. 3 diets based on corn, soybean meal, and canola meal were formulated. The first basal diet was formulated to contain 0.23% phytate-bound phosphorus, whereas the second and third basal diets were formulated to contain 0.29 and 0.35% phytate-bound phosphorus respectively by increasing the concentration of canola meal at the expense of soybean meal and cornstarch. 12 additional diets were formulated by adding 500, 1,000, 2,000, or 4,000 phytase units of a novel consensus bacterial 6 phytase variant or PhyG to the 3 basal diets. In addition, 3 reference diets were formulated by adding a commercial Burriozella phytase or PhyB at 1000 FTU per kilogram to the three basal diets containing no PhyG. A total of 144 growing pigs with an initial body weight of 12.7 kilograms were randomly allotted to 18 diets with 8 replicate pigs per diet. Pigs were assigned to treatment groups using a randomized complete block design with 4 blocks of 36 pigs and 2 replicate pigs per diet in each block. Pigs adopted to the diets for 15 days, and fecal samples were collected on day 16 to day 19, and on the last day of the experiment, pigs were euthanized and the right femur was collected immediately. Diets and fecal samples were analyzed for minerals to be able for us to calculate for mineral digestibility, whereas femurs were defatted, dried, and then ashed at 600 degrees Celsius for 16 hours. Data were analyzed in a 3 by 5 factorial arrangement with the pig as the experimental unit. The model included the amount of phytate-bound phosphorus, phytase, which is PhyG, and the interaction between phytate-bound phosphorus and PhyG as the main effects. Contrast statements were used to determine the effects of PhyG at 1000 FTU per kilogram on mineral digestibility and bone ash by comparing the diet with a diet supplemented with 1000 FTU per kilogram from the Burriozella phytase. Moving on with the results, this graph shows the digestibility of calcium in experimental diets. The x-axis represents the dose of the novel phytase, whereas y-axis represents the response parameter. Blue line represents the diet containing 0.23% phytate-bound phosphorus, orange, the diet with 0.29% phytate-bound phosphorus, and the green line represents the diet with 0.35% phytate-bound phosphorus. No interaction between phytate and PhyG was observed for calcium digestibility. Diets containing 0.35% phytate-bound phosphorus had reduced digestibility of calcium compared with diets containing 0.23 or 0.29% phytate-bound phosphorus. Inclusion of PhyG to diets increased the apparent whole tract digestibility of calcium in all experimental diets. An interaction between phytate and PhyG was observed for phosphorus digestibility, where PhyG increased the apparent whole tract digestibility of phosphorus, but to a greater extent in diets with 0.23 or 0.29% phytate-bound phosphorus than in diets with 0.35% phytate-bound phosphorus. The observed improvement in calcium and phosphorus digestibility upon PhyG inclusion indicates that the novel phytase was able to hydrolyze the calcium phytate complexes as well as some of the ester bonds between phosphorus and the inositol ring of phytate. The observation that diets containing higher levels of phytate had reduced digestibility of calcium and phosphorus indicates that a significant amount of these minerals are bound to phytate, and this increases the proportion of unavailable minerals for pig utilization in diets with higher levels of phytate. Inclusion of PhyG to diets increased bone ash, but to a greater extent if there was 0.35 or 0.29% rather than 0.23% phytate-bound phosphorus in the diets. This is likely due to greater concentration of apparent whole tract digestible phosphorus that was liberated from phytate present in diets containing 0.35 or 0.29% phytate-bound phosphorus. In this experiment, we also wanted to determine the efficacy of PhyG by comparing it with an established commercial Burriozella phytase. Blue bars represents PhyG, whereas orange bars represents PhyB. Here we can observe that at 0.35% phytate-bound phosphorus, diets containing PhyG had greater calcium and phosphorus digestibility compared with diets supplemented with the Burriozella phytase. Diets containing PhyG also had greater phosphorus digestibility in diets with 0.35% phytate-bound phosphorus compared with diets supplemented with a Butyrosella phytase. And overall, PhyG increased phosphorus digestibility more than the Butyrosella phytase. This indicates that the novel consensus phytase was more effective in liberating the phosphorus that is bound to phytate. PhyG was produced via fermentation with a fungal production strain, and it was demonstrated from previous research that the novel phytase had high activity in a wider pH range than the Butyrosella phytase. This indicates that PhyG could efficiently hydrolyze phytate and subsequently reduce the negative effect of phytate on nutrient digestibility. At 0.35% phytate-bound phosphorus, pigs-fed diets containing PhyG tended to have greater bone ash compared with diets supplemented with the Butyrosella phytase, indicating that greater phosphorus was available for bone mineralization in pigs fed the novel phytase. In summary, we have demonstrated that phytate reduced mineral digestibility, indicating that significant amount of minerals are bound to phytate, which increases the proportion of unavailable minerals for pig utilization. We have also demonstrated in this experiment that the novel phytase was able to efficiently increase the parental tract digestibility of minerals and also increase the bone ash of young growing pigs. The novel phytase also increased phosphorus digestibility more than the Butyrosella phytase, which can be attributed to its high activity over a wide pH range and subsequently reducing the negative effect of phytate on nutrient digestibility. We would like to take this opportunity to acknowledge DuPont for their inputs and financial support, and also to the whole team of Stein Monogastric Nutrition Laboratory. Thank you for your interest in this presentation, and please feel free to visit our website at nutrition.ansai.illinois.edu. Thank you.